Hello everyone, my name is LucaPegoraro from Royal Botanic Gardens Kew and Queen Mary, University of London, and I will talk to you about monitoring pollinators on Senecio doronicum in the European Alps. To start us off, some background on the plant family that I'm working on is the Asteraceae, also called the composite family because of the modular or composite nature of their inflorescence that comprises up to two different kinds of flowers, ligulate or ray florets on the outer rim of the inflorescence and disc or tubular florets in the center. They come in a variety of different floral shapes, and Asteraceae is one of the largest plant families in the angiosperms, and especially the montane and alpine elevation ranges. They make up to 50% of the total biomass together with the grasses, so they are extremely important in high elevation environments. A recurring theme throughout this talk is polyploidy. Polyploidy is the occurrence of two or more sets of chromosomes within the same cell, and especially recent polyploids can be looked at species in the making. When we look at sympatric mixed ploid populations, so populations that have two or more cytotypes, two or more ploidies together in the same place, we are effectively looking at a natural experiment that is ideal to test the effects of polyploidization. The polyploidization event is largely a microevolutionary event that played over long periods of time can account for the macroevolutionary pattern that we see, giving rise to at least partially the diversity of land plants that we see today. Polyploidy is one of the major drivers of diversity in flowering plants, and in fact, all angiosperms are thought to have undergone several rounds of polyploidization throughout their evolutionary history. One of the triggers that can enhance the chance of polyploidization is short growing seasons and low temperatures, and both these conditions were met during the glaciations, the most recent of which was around 10,000 years ago in the Alps. In this map you can see the maximum extent of the ice in the black outline and the glacial refugia in blue and red. Specifically, in the bottom left part of the map, there are the Maritime Alps where we set up our study site, and where there are glacial refugia, there is a higher chance that a new race that has arisen as an effect of the glaciation meets with a previous ancestral race, giving rise to sympatric populations. So our study site is Tête Grosse, that is a mountain in the region of Alpes de Haute-Provence in southwest France. It's 2,000 meters high, and it has high geographical prominence in the middle of the valley, and like many of the Maritime Alps, there is a very pronunciated difference between the exposed and non-exposed sides of the mountain. So bottom left, we got the south versant that is grassy and meadowy looking, whereas the exposed versant that is on the northern side, in this case, is mostly rocky and has much higher slopes. Our study species, as I mentioned, is Senecio doronicum. That is a fairly common Asteraceae found throughout mountain ranges in Europe and southern Scandinavia. It is mostly common in the octoploid variant, that is the most widespread, that flowers early in the year for alpine species, between mid-June to mid-July. And it also comes in a tetraploid variant that is much rarer and tends to occupy more marginal habitats on rocky slopes, and it flowers slightly later in the year, a couple of weeks later than the octoploid. So we did a study surveying the cytotype of Senecio doronicum throughout the Alps, and as you can see, the tetraploids are much rarer, and in fact the only population that we were able to find in which octoploids and tetraploids were growing together was in southwest France in Tête Grosse, and that is why we decided to carry out our study there. One of the first things that we did in this population was to look at the spatial distribution of the individual plants of each cytotype. So in blue, the blue points represent the octoploids, and the red points the tetraploids. Immediately you can see that the octoploids are much more numerous and occupy a much larger area of on the mountaintop, and the tetraploids tend to occupy a marginal habitat on exposed rocky slopes And also they don't tend to mix much with the octoploids. So already we can see that there is a microhabitat preference, and these cytotypes have a degree of spatial separation. As I mentioned before, these two cytotypes are phenologically differentiated, so they flower at slightly different times, but there still exists a period of overlap in which both the octoploid and the tetraploid cytotype were flowering together. I should mention that at this time, up to 20% of the plants that we analyzed had capitula, so had flowers fertile in this period. So the possibility of exchanging pollen was that there was a possibility of exchanging pollen between these two. We measured a bunch of different morphological traits on these plants, and even though the differences were not staggeringly evident, in fact it was sometimes difficult to tell in the field which one was of which ploidy level. Overall, on average, these plants were different in almost all the morphological measurements that we did. The most interesting ones to look at are the diameter of the capitulum, all the way to the left, and the height of the stem, that is the narrow box plot in the middle. And both of these measures are higher in the octoploid, so they have larger capitula and they are higher. But the number of capitulum, which is the one before last box plot, is the number of capitulum that is higher for the tetraploids. So tetraploids tend to be shorter, have smaller flowers, smaller capitula, but they have more capitula. One of the things that I would like to point out between these two morphologies is that the ligulate flowers that are the asymmetrical, more evident and larger flowers that are on the outer rim of the inflorescence, they have similar distributions between the two cytotypes, so on average they are similar in number. However, the tubular florets, that are the ones that sit in the middle and are much smaller, tend to be much more numerous in the octoploids. So this is an example of modularity, and octoploids are able to fit more tubular florets inside the same number of ligulate florets, and this is thought to be evolutionary strength of Asteraceae that are able to modify independently different parts of their body plan to achieve different combinations of phenotypes. We inputted this data into a principal component analysis to individuate the main axis of variation, and indeed we found that the height of the stem and the number of tubular florets were the variables that most differentiated these two cytotypes. Another thing that's apparent is that octoploids occupy a larger morphological space than tetraploids, and they also tend to encompass most of the variability found in tetraploids. So this was another interesting point emerging from the phenotypical analysis. Another thing that we noted was that the reproductive success between the two cytotypes was widely different, with tetraploids having many more viable seeds per head than octoploids. We think that this is at least in part due to the fact that tetraploids, even though they were more numerous and in general the population produced more capitula, they suffered a much higher predation rate by a predispersal seed predator, we think a weevil beetle of the Curculeonid family, and this impacted gravely their reproductive output for the season. If you notice, the proportion of predated capitula for tetraploids is much lower. Before moving on to the pollinator monitoring part, I should mention that together with Sarah Barlow, we published earlier this year a review on automated pollinator monitoring system, going over all the pros and cons of the different methods available today. But we used the RANA pollinator monitoring system, and that you should be familiar with by now. Our particular equipment was made up of a 720p autofocus webcam, an Odroid data logger, and 110 ampere lead acid car battery. So due to the nature of the alpine site that we're using for our population study, we had to meet some logistic challenges. So especially we needed a lift by a local workman to carry our equipment to the top of the mountain, which is 1.5 hours to 2 hours away from the nearest road. So we carried our equipment there, we deployed the power source, the battery, and the data logger inside a clear plastic box covered in a white tablecloth and a lid to prevent the sunlight as well as rain to get in. And we secured the cameras on a bucket full of stones. So sometimes we had to secure that with a metal rod to prevent the buckets from rolling down to valley due to the steepness of the slope. Here I'm gonna play a clip of some pollinators monitoring footage that we collected. You can see that the time signature is skipping because the system is suppressing the shadows and movement of the wind of the plants. Here are some visitors, typical visitors of this plant. These are mostly hoverflies. This is an Eristalis that is visiting. One thing that you can appreciate from this footage is that you get many more angles on a single visit of a single visitor, so it's much easier to get a confident identification, as well as appreciating the behavior. For instance, here you can see the mouthparts going over each single floweret, probing each one individually. As for most Asteraceae that have disc-like inflorescence, like Senecio doronicum, we expect this to be pollinated by a variety of insects with short mouthparts, but there are in fact other pollinators with longer mouthparts sporadically visiting. Here is a rundown of the most common pollinators found on these that we found on this Senecio doronicum. They are mostly hoverflies, and Eristalis tenax and Chrysotoxum intermedium were particularly abundant, but as well as solitary bees like Lasioglossum, and every once in a while we also got other orders like Lepidoptera with this Lasiomatomera visiting. During the monitoring period, we also collected insects during the flowering time of these two cytotypes, and you can see on the column on the right that the three main colors, blue, red, and green, are equally divided. So this means that the butterflies, bees, and flies were almost equally abundant at the study site. But if we look at the feeding visits on Senecio doronicum, we can see that the flies are much more abundant than all the rest and make up the most of the visits, as well as bees making up a significant part, but butterflies tend to avoid visiting this plant for the most part. I should also mention that the floral resource context in which these two cytotypes bloomed was widely different, and in fact octoploids had many more flowers of other species available to pollinators to choose from than the tetraploids, which grew in a much more impoverished community. One thing that I noticed looking at the footage was that pollinators tended to spend more time visiting octoploid inflorescences, and if you remember, the octoploids were the ones that had a higher number of tubular florets, so the pollinators tend to spend more time on inflorescences that are richer in flowers, that have more flowers, and I thought that was an interesting point to notice. The table presents a rundown of the percentage of visits, and as you can see, the number of the percentage of nectar feeding visits is much higher for the tetraploids than for the octoploids. Another thing that was different between the two cytotypes was the hourly visitation rate that was higher for the tetraploid, and this accounts for the overall visits number that is much higher for the tetraploid overall. One thing that we can notice as well is that the column on the right, that are the feeding visits on octoploids, is dominated by the dark greens that correspond to the Cirphus genus within the hoverflies, that is one of the main pollinators of the octoploid cytotype of Senecio doronicum, whereas the tetraploid cytotype is dominated by the light green, that is corresponding to Eristalis, that is the main pollinator of tetraploids. Here I present an MDS analysis of pollinators community. So in the polygon in red encompasses the pollinator community visiting the tetraploid, and Eristalis has a prominent position within this community. And in the blue polygon is the octoploid pollinator community, with Cirphus making up most of the visits. One thing to notice is that these two polygons don't even touch each other, so the pollinator communities would seem to be largely separated and different from each other. Running a dispersion analysis on these communities, we can see that not only the extent of variation is similar, but also they barely touch each other. So I am confident in saying that the pollinator communities between these two site types is largely different. And to my knowledge, this is one of the first times that it's been reported that a generalist plant has specificity in their pollination, their pollinators. Um, this level of detail is only possible with these new automated pollinator monitoring techniques that are emerging. And one of the other things that I would like to look at in the coming months is to model the temporal visitation preference of pollinators, because looking at hours and hours of pollinator monitoring footage, I have the feeling that different genera of hoverflies, for instance, tend to have different preferences of when during the day they are visiting, and this can have important consequences on the efficiency of pollen transfer between plants. So, and with this, I conclude the talk. I should thank my principal advisors as well as many other collaborators and institutions that helped me throughout my journey. Thank you for your attention.